So, Dr. Robin, I think first time is for you and Dr.
Yeah.
Aradhana also.
We can have the slides. We can start now. Yes, so host has muted everyone. We are starting now. Welcome to the 256th edition of Thursday Musings. We are hoping to get a grasp of genetics in neurosciences and taking the sessions further in some other parts also. So welcome all of you. The session is being recorded. The recording will be put on the YouTube channel. The questions are invited in the chat box. So you should type your questions in the chat box. They will be taken up by the speaker. Next slide, please. Professor Dr. Tufan Pati sir is the chairman of this program. Sir, handing over to you. Please take over.
Tufan. Thank you very much, Dr. Adim. Good evening, all participants, and best wishes and eve of Teachers' Day to all. And I take the privilege to congratulate the vibrant moderators of this series, Dr. Amrit Patajoshi, Professor of Psychiatry, Dr. Alim Siddiqui, Professor of Psychiatry, Dr. Shobit Gar, and Dr. Vishal Chavra. They are vibrant moderators who are the mainstay of the program. And with us, we have a chairperson, Dr. Raveen Goyal, MBBS RGHS Bangalore, DPM FMC Pune, DNB Deva Institute Varanasi, working as head psychiatry in Department Kushubhav Thackeray Hospital, Rewa, Nodal Officer District Mental Health Program, Rewa, MP, Vice President Indian Psychiatric Society, MP Chapter. Welcome, Dr. Raveen. And I have the pleasure to introduce Dr. Sai Krishnatika, MBBS DPM MD PDF Neuroclinical Research, current designation Additional Professor in HOD Psychiatry, AIMS, BB Nagar, Hyderabad. Awards and honors Tilak Venkavara Award 2024, Mafate Award 2020 and 2024, Young Psychiatrist Award 2018, and Bhagwat Award 2016. Editorial Board, Asian Journal of Psychiatry, Indian Journal of Psychological Medicine, Publications, Books, Chapters 4, Chapters in Books 36, PubMed Journal Articles 1 and 7, and H Index 24. Areas of interest, psychotic disorders, cognitive neurophysiology, and brain stimulation. Welcome, Dr. Sai. And with this, I hand over the meeting to the chairpersons. Dr. Sai will be introducing topic. Dr. Robin, please introduce your speaker, which will be displayed on the board. Over to the chairpersons.
Thank you so much, Tufan sir, and welcome all of you. We have an esteemed speaker with us. Dr. Aradhana will be speaking on a field which is called as neurogenetics. So neurogenetics is a field where we understand how genes interact and interact with the development of neural system, and also how it modifies different neurological disorders, psychiatric disorders. I mean, ma'am is also going to talk to us about what different techniques we have for understanding these genetics. I think, like we've introduced earlier, that this is going to be a series of lectures from Dr. Aradhana. Today's lecture will be focusing primarily on the basics of genetics. She may not be jumping into directly the basics of genetics of psychiatry, but then basics of genetics here. Without much ado, I request Dr. Robin to introduce ma'am.
Yeah, thank you, Tikka sir. First of all, thank you organizers for giving me this opportunity to be here. Every Thursday I used to think that I have to attend this meeting, but thank you for making me a chairperson, so I'm definitely there in the meeting. And it's an honor for me to introduce Lieutenant Colonel Aradhana Dwivedi ma'am, because she's also from AFMC, and I learned my psychiatry from AFMC, so I know that how much she's been grilled to complete her MD from that institute. So she is the first medical genetist from Armed Forces Medical Services, because yes, I didn't know that there is anyone, but definitely she is the first one. She is currently working in the Command Hospital, Southern Command, Pune, and alumnus to the prestigious Armed Force Medical College, Pune. She has got numerous publications in various domestic and international journals, and her area of interest is parental and cancer genetics, lysosomal storage disorders, dysmorphology, inborn errors of metabolism, neuromuscular disorders, and neurodegenerative disorders. She's a member of Indian Academy of Pediatrics, Society of Indian Academy of Medical Genetics, American College of Medical Genetics, and so many genetics. Sorry, ma'am. This area is actually, I think it is new for everyone who is there in this presentation. Her current research projects are inherited disorders of brain, heart, and kidney. And she's been working as a genotype-phenotype correlations in patients with renal tubular acidosis. It's an ICMR project. Study of diagnostic yield of whole exome sequencing in patients with CAKUT, congenital anomaly of kidney good. So yes, without wasting much of a time, because I think whole presentation will occur for her introduction. So ma'am, over to you. Please enlighten us with this such an interesting topic. Thank you.
Thank you so much, Dr. Robin, and then great to learn that you also learned psychiatry from FMC. And we'll just move on to this. The slides are visible, I hope.
Yes, ma'am.
The slideshow.
Yeah, I put it on slideshow. So thank you so much. At the outset, thank you to Dr. Shobit Gar, who reached out to me and made me part of this Thursday musings along with the whole team. Looks very interesting team. It's my first interaction, and I said that because it will be too difficult to cover so many things in one go, so we may have some series for 2-3 lectures where we can these Thursday musings where we cover the genetics which is actually pertaining to psychiatry. And today we'll be just trying to learn the basic terminology and the testing which are there at present in the armamentarium, and then how to utilize it later we can see. So if we look at the field of genetics, it's a very young discipline. It was recognized as a medical specialty only 3 decades ago. It was always more important for families who were affected with genetic disorders. But then with the completion of Human Genome Project and introduction of the next generation sequencing in the armamentarium, the game has changed. Now it is available at door-to-door services. So some of the important landmarks which this field has seen starts from 1665 with the discovery of cell, then the identification of the chromosome in 1885, then identification of the correct number of human chromosome in 1956, then the introduction of Sanger sequencing method in 1977, then the Human Genome Project started in 2001, and then it got completed in 99, and then got completed in 2003. And then in 2008, we got the platforms for next-gen sequencing, and 2011, the next-gen sequencing came into the clinical practice. Since then, there is no looking back, and we are only moving forward, and we are in the NGS era now. So if I may, I always ask these questions to the audience. Can you think of any condition, any medical specialty which is devoid of genetics? Actually, none. So all the, whether it's a psychiatry or neurology or gynae, pediatrics, wherever you say, cancer, oncology, everywhere, every condition has an underlying genetic. So all these things can be viewed through genetic lens. And obviously in a neurosciences it plays a major role, because starting from the brain development, your synaptogenesis, then coming, which leads to your personality formation, your behavior genetics, and various neurodevelopmental disorders, genes have got many roles to play, which we can talk in our subsequent subsequent talks. So when somebody asks why we should be offering genetic testing, because some of the old thoughts goes that no, these are the rare disorders, why should be offering genetic tests? What will you do by making the diagnosis? So first of all, let me tell you, because I am being a geneticist, though individual disorders are rare, when we look at the aggregate, they are sizable. And we need to have the diagnosis because some of the disorders are treatable. If you talk about the neurogenetics, maybe we are dealing with channelopathies, or at times some epileptic encephalopathies, and some, like there's a disorder which is cerebral creatine deficiency disorder, wherein you just supplement creatine and then you will see improvement. So some of the disorders are treatable. If we don't have the diagnosis, we can't start specific treatment. Definitely to provide the accurate recurrence risk, because once you have a child affected with the disorder, the family would want to know whether it is going to recur, and how much is the chance and what can be done about it. And if we don't have a diagnosis, we can't provide prognosis, whether it is going to be static problem, it is going to progress, or it is going to improve. And last but not the least, every family, every person wants the closure. So and maybe it's just end the diagnostic odyssey the family is going through. by providing the genetic testing. For any specialty, we say, when do you suspect a genetic disorder? When does it come to your mind that maybe I'm dealing with an underlying genetic problem? So these are the 4 broad groups that whenever you are seeing a person who has got dysmorphic features, which are very evident on physical examination, constellation of the multiple anomalies on one patient, and then there is unexplained neurocognitive impairment, or the family history which is suggestive of a hereditary disease. These are the 4 pointers for any broad. I'm just talking about the broad subspecialties. Then you will be thinking that maybe you're dealing with underlying genetic disorder. So let's look at the organization of human genome first to understand the genetic testing and further. So the genome means the sum total of DNA which our body has, and most of this DNA is in the nucleus. A small amount of this genome is also in the mitochondria in humans. In the nucleus, this genome or the whole DNA is concentrated in 23 pairs of chromosome. And what is a chromosome? Chromosome is a coiled DNA. If you uncoil it, you will find a single DNA molecule. And then what is a gene? Gene is that part of that segment of DNA which is making a protein. And the gene has a coding portion which is called exon. It has a non-coding portion which is called intron. And besides that, gene also has promoter and regulatory region to control its further functioning. So this is the basic structure of human genome which we have. And now based on this structure, we can have various mutations which we call variations or variant now. So this variation can be at the chromosomal level or can be at the nucleotide level. So at the chromosomal level, we can have a problem with the numbers, means we can have a numerical abnormality. We can have, because we know we have a 46 pairs or 23 pairs of chromosome, which makes 46, and that is 22 pairs of autosome and 1 pair of sex chromosome, XX in females and XY in males. So if you have a number problem, number could be exact multiple of n, that is haploid number. Somebody may be having 23 is 46, then 3n is 23 into 3. So those are called ploidies or polyploidies. Polyploidies are generally not viable, so these babies will not be born. Then we have got aneuploidies. Aneuploidy means which is like your Down syndrome, extra chromosome 21 or 13 or 18. So these may be viable chromosomal abnormalities. So when these are the numerical problems, then we have the structural variants. Structural variants could be your translocations, repeats, or inversions. Then just remember the copy number variants, because this is a term which we'll be using and will be coming across in most of your literature. So the copy number variant is something which is basically DNA segment which is duplicated or deleted compared to the reference genome. When it is deleted, there is a loss of the copy number. Usually this copy is more than 50 base pairs of the DNA. And when there is a duplication, there is a gain. Then now coming to the second part, the variations or the mutations at the nucleotide level. Nucleotide means we know we have got these 4 bases: adenine, guanine, cytosine, and thymine. So when you look at the single SNVs, so here there is a copy number variant and its counterpart is SNV, that is single nucleotide variant. So in a single nucleotide variant, you can have most common is substitution. A is replaced by T. So that or maybe A is deleted or if T is inserted. So these are the single nucleotide changes. This one base pair change is happening, whether inserted, deleted, or substituted. Or maybe small indels. Small means 2 to 3 base pairs are deleted or inserted. Those are called. So these things are happening at the nucleotide level. So based on this variations, we can have, we can broadly divide genetic disorders in 3 categories. That is chromosomal, monogenic, and polygenic. As I said, chromosomal because of the change in the number of chromosomes or structure of the chromosomes. And this change in the number, as I said, could be like Down syndrome, number is increased, or at times it's a monosomy like Turner syndrome, where there is only one X chromosome, or we can have structural problems. Then coming to the second major group of disorders, that is monogenic. Monogenic means single gene disorders. Most of your childhood neurodegenerative disorders, where all those Batten disease, etc., all these leukodystrophies primarily. So these are monogenic because there is a mutation in a gene which is responsible for the disease. And these are the group of disorders which follow Mendelian patterns of inheritance. When we say the disorder is inherited in a recessive manner or dominant or X-linked, we are referring to the single gene disorders. And the examples like Duchenne muscular dystrophy or spinal muscular atrophy. Duchenne muscular dystrophy is an X-linked disorder, while SMA is an autosomal recessive condition. And most of the classic neurodegenerative disorders in childhood are autosomal recessive in nature, and these are monogenic conditions. Then comes a third group of which is called polygenic or multifactorial. As a group, since I'm audience is a psychiatrist, group of psychiatrists, so this may interest them further, which I will dwell upon later. So the disorders like autism spectrum disorder, Alzheimer's, Parkinson's, etc. So here it's not a single gene which is present in a person and can cause the disease. It's about multiple gene to gene interaction as well as gene and environment interaction. So it's a polyfactorial condition, somebody having an underlying genetic susceptibility, and there are some other factors which are required for the disease to develop. So this is the third group, that is polygenic. So these are the three broad categories of genetic disorders which we see in our clinical practice. Now, based on these categories, we have different types of tests to diagnose these conditions. So what are the tests? Tests could be cytogenetic tests, molecular cytogenetics, and molecular tests. So what is a cytogenetic? Basically, study of the chromosomes. And then conventionally, we all know it's a karyotype. Karyotype is the most old test. And after that, there were some limitations of karyotype. That's why we went into the molecular era. And to decode further zooming further, to know the further details, fine tuning those things. So though we are in NGS era, the karyotype is not yet dead. There are some indications wherein karyotype is only karyotype can help us. Like if you see a child, these children with Down syndrome, or there is a girl with Turner syndrome who has presented with short stature and learning disability, or in the middle there is a child who has cleft lip and palate. Basically, he has trisomy 13. So these are all chromosomal abnormalities, and these are all numerical abnormalities. So in these cases, only karyotype would suffice. We don't have to think of hi-fi test of exome sequencing, because here we are dealing with the chromosomal abnormalities. So karyotype is still indicated for your common aneuploidy assessment whenever there is indeterminate gender, delayed puberty, short stature, or some other recurrent losses, etc. Then the karyotype is still indicated. What are the benefits of karyotype? Basically, karyotype can visualize the entire genome. All 23 pairs of chromosome can be seen. Individual cells and individual chromosomes can be seen. This is the only test which can detect balanced translocations, inversions, or ring chromosomes. The limitation is that the resolution of karyotype is limited to 5 megabase pairs. If there is a micro deletion or duplication, it will not be picked up by karyotype, and karyotype requires actively growing source of cell. So in a dead cell, we cannot perform karyotype. So these are some of the limitations. To overcome some of these limitations, maybe some other tests were developed. So like FISH, you must have heard about the FISH test, fluorescent in situ hybridization. So FISH is a probe-based test. So we have to remember then whenever there is a test which is probe or the primer based, then it doesn't cover the whole genome. Then it is a specific, because FISH is performed for specific regions. I mean, if we have for that, we have to know the diagnosis. If I know I'm dealing with a child with a Williams syndrome, who has a deletion, small micro deletion in the 7q region, the long arm of the chromosome 7. We know the diagnosis beforehand or on the suspecting. So I will design the probe accordingly, and then which will detect that deletion, heterozygous deletion in 7 region. So this is a limitation, though the benefit is that you don't require actively growing cell because it is done in the interphase. And secondly, it can turn almost any DNA into probe and very short turnaround time. Karyotype needs culturing. It takes about 10 to 14 days for the results, while FISH results are available within 48 hours. So these are the benefits, but the limitation is that it doesn't visualize entire genome, unlike karyotype. And then these are some of the utility of FISH, like in Williams syndrome on your left and your right, we can see a child who is diagnosed with 22q11.2 deletion, that is commonly called DiGeorge syndrome. So these are the tests where you'll be needing FISH can be utilized. Then FISH is slightly more tedious process and more costly. So we have something better test called MLPA, that is your multiplex ligation dependent. So as I said, just remember copy number variant and single nucleotide variant. When we say about the copy number variant, basically we are comparing it with the reference human genome. in which compared to that, the segment is either duplicated or deleted. And there is a considerable number of base pair, usually more than 50 base pairs are deleted or duplicated. And these are the large deletions and duplications, which are called copy number. So in MLPA, we are using multiple probes, but not genome wide. I mean, it doesn't cover the complete genome, but then yes, multiple probes are used depending on the condition we'll be using. But remember that it will not detect point mutations or what we call single nucleotide variant. And if there is a balanced translocation, which can be picked up by karyotype, it cannot be picked up by MLPA. So what are the common here? This is a child with calf hypertrophy and then difficulty in getting up. So basically this child was diagnosed clinically to have Duchenne muscular dystrophy, one of the most common muscular dystrophies. And the child on the right is clinically diagnosed to have SMA, spinal muscular atrophy. And these three sets of children who are obese are basically suspected to have Prader-Willi syndrome. So when we are dealing with these, like if in this example, the 5-year-old boy who presented with obesity, hyperphagia, developmental delay, there was feeding difficulty and hypotonia in infancy. So we are suspecting Prader-Willi syndrome based on this clinical history. And whenever you're suspecting Prader-Willi, your first test of choice would not be karyotype, would not be exome sequencing, it would be your methylation studies, which are done through MLPA, or we call MS-MLPA, that is methylation specific MLPA. So because methylation leads to gene silencing in particular region, and that is causing the problem in the Prader-Willi. So we have, based on what problem we are dealing with and what we have to know, which test is the best suited for that condition. Similarly, for spinal muscular atrophy, which is a very common condition in India, and 1 in 38 out of will be carrier of this condition. So for the carrier status, and now it has been advised that every couple, whoever is planning conception, should be tested for carrier screening, because carrier frequency is very high in our country. And how to go about the testing? The test of choice is again MLPA, because in this condition there is a deletion of exon 7 and 8 of SMN1 gene. So it's not a single nucleotide change. In 95% of the cases, the deletion is in the exon 7 and 8, which will be picked up by MLPA. So when we are suspecting those conditions, and remember that whenever we are doing exome sequencing also, and we are finding one variant only. Suppose we are dealing with a disorder which is autosomal recessive in nature, and autosomal recessive should have 2 variants. Ideally, biallelic, we call it, one on each chromosome. So if we find only one variant and our suspicion is very strong, then in that case, we need to review at the literature that maybe in that gene are the large deletions or duplications known. If that is known, then we may go for MLPA or the microarray to look for the copy number changes, which may be another mutation which is there in the patient, which is not being detected by exome sequencing. So this would be the utility. Similarly for DMD, because DMD, 70% of the cases are because of large deletions and duplications. So the first test of choice would be MLPA for DMD again. After MLPA, we come to another test which again detects copy number variants, and that is your microarray. You must have heard about microarray, because this is something which, as a psychiatrist, a lot of children come to you, autistic patient. So this is the test of choice for autism. So what is the advantage of microarray? It is a whole genome coverage. Like karyotype, it also visualizes all 23 pairs of chromosome. It detects copy number changes, but then advantage of this over karyotype is that it can detect at much higher resolution. It can detect up to 40 kV deletions, and so that micro deletion and duplications are implicated in many neurodevelopmental disorders, and that can be picked up by microarray. But unlike, as I said, karyotype is the only test which can detect balanced translocations. Usually balanced translocation doesn't have phenotypic implications, but then they can have reproductive issues later on. So, but the microarray will not detect that. And whenever we are doing a test which is covering whole genome, remember that we may get some information which is not clear or which is unclear. So those things need to be told in the pre-test counseling, because we may be surprised to get something which we are not expecting, because this is something which covers the whole genome. So these are some of the disadvantages of whole genome, doing something which covers the whole genome. So what is the current indications of chromosomal microarray? For all, it has been recommended by American College of Medical Genetics as the first line test for unexplained intellectual disability or cognitive impairment, children with autism spectrum disorder, when there is a multiple congenital anomalies, and for the prenatal diagnosis also with anomalous fetuses. So these are the 4 indications which are where in the first line test for CMA is the first line test. Look at this case scenario. This baby, 22 months old female toddler, she came to us with the delayed milestones, failure to thrive. There was a dysmorphism and there's a congenital heart defect. Whenever you see all these, maybe there's a, just to know a little bit that how do you go for syndrome search. So delayed milestones are seen in many neurodevelopmental, so that is the main problem actually in these things, and that may depend which domain is more affected or equally affected. So all those you look for, if there is a particular clinical handle, there's a particular dysmorphism which is noted, then you can put your clinical handles in OMIM, that is online Mendelian inheritance in men. So you can put all those clinical features. Then we have got LMD, London Medical Database. Then you got Phenomizer and POSSUM. POSSUM is not free of cost, but other 3 are free of cost. So you can put and then maybe see what you're dealing with. It will give you some diagnosis. Maybe it's some micro deletion or maybe some single gene disorder, because that would help you to know that whether you are dealing with which test you should be offering first. So then we did that. In that case, we did not find any there was not a single gene disorder, but there were some multiple micro deletion syndromes which were coming in the syndrome search. So we went ahead with the chromosomal microarray, and this is the typical report of a chromosomal microarray. The first thing we on left, you see CN state means copy number state. Copy number state in a normal individual we will see for any chromosome, that will be 2. So if it is 3 means it is a gain. If it is 1, it means loss. Gain means 1 copy gain. So it's a heterozygous duplication. Then on which chromosome? It is a 16 chromosome. Where exactly in chromosome 16? On the long arm of chromosome 16, that is Q, and the bands are 2.1 to 2.4 because chromosomes are divided into bands and subregions. And what is the size of this gain? The size of this gain is almost 30 MB. And the total number of genes in this gain is 360. So the OMIM genes, in this gained part, duplicated part, there are 360 genes. And as per the classification, there are various databases and resources which classify this gain at chromosome 16 as a pathogenic. So this is basically the child is having partial trisomy of 16 chromosome. As I said, if you say the full trisomy of 16 will not be viable. So partial trisomies and monosomies can be viable, and they can present with various neurodevelopmental problems, dysmorphic issues, etc. So this is how we are dealing with a 16q duplication. Since most of you are familiar and you keep seeing autism spectrum disorder, so let's talk about this, because this is when we look at the CMA is a test of choice, the first test for the autism spectrum disorder for a non-syndromic autism. When you're a syndromic autism, then that would differ because that depends on case to case. For a non-syndromic copy number variation, as I said, autism is a multifactorial condition. There is environmental, epigenetic, double hits, and sex-linked modifiers. And there are copy number variations which can be found in about 15% of the patients with autism spectrum disorder. And if suppose, as I said in the previous example, there was a 16q duplication, and there were 360 genes, but all the genes would not be responsible for that particular phenotype or particular clinical feature. In that also, there would be a particular gene which we express more in the brain, and then that might be causing a problem. So based on that, these copy number variants, these are some of the genes which are implicated in autism spectrum disorder, like SHANK1, SHANK2, 3, and other NRX1, SCNA2, etc. So we have to see in that the duplicated or deleted region, which is the particular gene which may be causative of the phenotype which we are dealing with. So, and how the genes are working, these genes which are implicated in various neurodevelopmental disorders and autism spectrum, they are interfering with the metabolism, glucose metabolism, your monoamine metabolism, neurotransmitter metabolism, and these leads to various cognitive, behavioral, and social abnormalities which are observed in patients with autism spectrum disorder. So this is the flow chart for the diagnosis of autism using microarray. When you are using microarray, there are 3 scenarios. Either it is normal, or it is abnormal, or somewhere in between we are finding something called variant of uncertain clinical significance. So normal, if it is normal, then you have to do other testing. It's the male child, then you go for Fragile X testing. Maybe the all obvious dysmorphic features may not be present, but it is pertinent to go for Fragile X testing. Or maybe it's suppose the female child is a stereotypic behavior with autism, microcephaly, you may consider MECP2 testing for Rett disorder, or depending on the underlying clinical diagnosis, you can consider other molecular test panels. And if you're dealing with an abnormal, which is like pathogenic, then you do the parental testing to confirm it's a de novo or it is a inherited. Mostly it would be de novo if the parents are usually normal, and the de novo would signifies more of a causation. And the for again variant of uncertain significance is something which needs to be further studied, and again your parental segregation is required. And these VUS, maybe it's, as I always say, it's a dynamic thing. VUS, if you find a VUS that is variant of uncertainty today, it may not remain VUS tomorrow. Tomorrow, with the as we gather more evidence, it can shift towards benign or it can shift towards pathogenic. So we have to look at the reports and we have to reclassify variants again and again, depending, and then it may change its significance over the time. So now we have spoken about microarray, we have spoken about the cytogenetics and we have talked about the molecular cytogenetics, that is your FISH, MLP, and microarray, which are primarily looking at your CNVs, that is your copy number variant. Now let's look at the nucleotide level. When nucleotide level means the DNA is there, we have got the DNA. In the DNA, these are the bases which are arranged. We know, we remember from our biology classes, ATG is the methionine code, which is the start codon. So then in every gene, the gene is the segment of DNA, and every gene starts with ATG, your start. And after that, at what number is G, C, T, T, depending on the various genes. And we have the structure of these genes thanks to Human Genome Project. So we know now, so decoding, sequencing is nothing but decoding the sequences in that gene, at which particular position, which particular base pair is there. So that is called sequencing. And then by this, we know, since we have a reference genome, the reference sequencing, we know that in the patient where it has altered. So now we have got two groups, first generation sequencing and next gen. All of you know, must have heard about NGS. You keep hearing about NGS here and there. So the first generation sequencing was something which was devised by Sanger, Frederick Sanger. And along with there was another method which was called Maxim Gilbert, which went out of vogue. And the Sanger sequencing remains gold standard for years long, and still it is. So these are the first generation. After that, and problem with the Sanger sequencing was that it's tedious. It was initially manually, then automated, but you can only sequence one gene at a time. So it takes, it's a very it's a lengthy process. Then we came to the next year. After that, the platforms were developed, and platforms wherein the high throughput massively parallel means multiple samples you can put together, and the multiple genes, the whole genome of the human beings or other animals, also smaller animals, can be sequenced. So that is the advantage of next generation sequencing. So the principle of NGS is high throughput massively parallel sequencing. And then we have got some other molecular tests which we'll see. So coming to we have spoken about the first generation, and then after that came the second, third, and fourth. Second and fourth all are part of the next generation, and they are using different platforms to sequence the human genome, to our human genome, basically. And now how do we utilize this? Let's see what are the roles of Sanger sequencing. Sanger sequencing can still be used for small genes, because I said it's a lengthy process, it's a tedious one, so it can not be implicated. Since now we have NGS, for the larger genes, we would not like to go for Sanger sequencing. Then if the gene is large, but you know there's a particular hotspot mutation, then you have to only look for that mutation, then also Sanger can be done. Or when you have an NGS result and you want to validate your findings, then Sanger should be employed. But remember that whenever there is a primer, here in Sanger, we are designing a particular primer for that gene. So your diagnosis should be known beforehand. So these are some of the examples wherein, like, maybe just to know, I know it may not interest you much, but then thalassemia is one of the commonest condition and a small gene where we are doing Sanger sequencing. So this is the result of the Sanger sequencing for any condition, because these different colors are different base pairs. T is red, G is green, G is black, A is green, and C is blue. So by this, we know at which position there is a change, and there is a base pair has been added, deleted, or changed, substituted. And then we see the mismatchment, as you can see in the picture where the red circle here, the G is replaced by T here, and the subject and the query means there is a query of the patient and subject is a reference genome. So we know there's a mutation, and at this position, we are using a standard nomenclature to define these variants or mutations. So after Sanger, if we see these group of patients, one patient is having the diagnosis Bardet-Biedl syndrome, where you see intellectual disability, renal dysfunctions, eye problems. Why I'm wanting to show, because there's a condition where there's more than one gene. Like 20 genes can cause this condition. And this lady has adenoma sebaceum, or we call facial angiofibromas. Basically, we are dealing with tuberous sclerosis complex. And on the right side, this man has multiple neurofibromas. So we are dealing with NF1. So again, NF1 is caused by a single gene, that is a neurofibromatosis NF1 gene, but then it's a very large gene. So it's not amenable to Sanger sequencing. Tuberous sclerosis is caused by 2 genes. So there's not a single gene here. And then, as I said, Bardet-Biedl is caused by more than 20 genes. So these are the conditions where we would like to go for. Some of the conditions, NGS would be better. So NGS is, as I said, massively parallel. And using this technique, what we can do, we can utilize this technique to sequence a single gene. For example, as I said, if I'm suspecting neurofibromatosis type 1, my diagnosis is clinically clear to me. So I would like to go, I have to see then which mutation this patient or the family is carrying. So you can use only a single gene testing by NGS method. Then we can go ahead and do have a group of genes. Suppose we are dealing with a muscular dystrophy, and there may be multiple few 20-30 genes. So we can design panels for the better coverage of that group. So that is panel. Then the clinical exome. Remember, exon is a coding portion and exome is a plural form of exome. And clinical means medically relevant, what is already known, the diseases and the genes which are already known, and it has been enlisted in OMIM, that is your online Mendelian inheritance database. And around the list is every day increasing because we are finding more and more genes and the diseases. So currently around 8,000 genes are there which have been implicated in various medical genetic conditions. So for whenever you are doing clinical exome, you can do for the conditions, again, your phenotype should be more or less clear to you. And the whole exome means all the coding portions of the gene, all the genes of 22 to 25,000 which our body has will be covered. And if you're not sure what you're dealing with the patients, it's a very non-specific. Suppose it's an intellectual disability, your unexplained ID, you have done microarray is negative, then there could be chances there is some G, because there are more 300 genes which can cause unexplained ID mutations in those genes. Then you go for exome and you may pick up the diagnosis. And this is the condition. In whole exome, you may find the new gene discovery also, because it is going to cover all the genes here. And then coming to the whole genome. Exome is a coding portion. When we look at the genome covers both coding as well as non-coding, because ideally the 85% of the human genetic disorders are caused by the genes mutations in their coding portions. Well, 15% can be a non-coding. So that is intronic regions. Whenever there's a strong suspicion, there's a strong family history, and we have exhausted other tests, exome, and we have exome is negative, your microarray is negative, we are still suspecting that yes, it could be, then maybe a intronic mutation may be hidden. In that case, whole genome will help us, and whole genome will also give an idea about the structural variations on copy number changes also now. So when we have found the variant, if I test someone, we do whole exome sequencing for someone, we'll have lakhs and lakhs of the variant. So, but all the variants will not be relevant. We have to do annotation. Annotation means, I mean, this is the workflow. Once you've got the data of any patient with, like, as I said, it will be more than 1 lakh variant in that. But then those 1 lakh variant are responsible for the difference in your eye color, your nose shape, etc., your hair color, but they are not actually disease causing. So actually something is disease causing, then how do you proceed? You look for the population data. Ideally, if something is disease causing, that variant should not be present in high frequency in population. If the variant present in high frequency in population, means it is a polymorphism. So these are some of the population databases, 1,000 genomes, your exact exome aggregation consortium, etc. So population database we look at the disease databases, like some of these databases are your ClinVar, HGVS. Then we have to look, there are some functional studies which has performed, maybe some mouse models, animal models have been performed, this particular mutation has been checked. And then you do the phenotypic segregation, means if there are multiple, 3 members are affected and 3 are unaffected in that family with the similar disorder, then if you're tested and all 3 are having the same and in another normal, this is absent, then it is segregating. We say it's a segregating in the family. And then we gather all these evidence, and based on the strength of evidence, we classify these variants into 5 classes: your benign, likely benign, uncertain significance, likely pathogenic, and pathogenic. So this is how we go about when we get the NGS raw data, and we annotate and then we classify based on that. So let's look at, as I said, that NGS is a method which can be employed to test your single gene, your handful of gene, or maybe 8,000 genes, all genes coding portion, or both coding and non-coding portion, that is your genome sequencing. So if we look at this example here, 13-year-old child who presented with ID and neuroregression, microcephaly and stereotypic hand movement. So when we saw her in OPD, all of us, those who have seen Rett disorder, they would not miss it. So she was a typical of a Rett disorder. And then when my suspicion is very strong, I know I'm dealing with Rett. And the most common cause of Rett disorder is MECP2 mutation in the MECP2 gene. So we went ahead and only sequenced MECP2. So this is how MECP2, and it confirmed the diagnosis. There is, so when you get this report, just for this understanding, so there's a gene and the transcript. Transcript means this is a reference transcript, because the gene can make many more than one transcript. That is called alternating splicing. So through alternating splicing, more than one transcript can be generated, and then the base pair position may change depending on which transcript is being used. So this is the NMID, which is your transcript ID, and using this reference ID, this has been shown that this particular variant which has been detected in MECP2 gene is located in the exon, that is exon 4 of the gene, and the particular variant is C.502, means at the coding portion, at the codon, as I said, the first would be C.1 would be ATG in any gene. And then at the 502 position, cytosine is changed into thymine. This is C2T. And this change is what problem it is causing at the protein level? It is causing arginine is terminated, means it is a stop codon. It is adding stop codon, and it is causing nonsense. It's a nonsense mutation because it is terminated. So the length, the protein is now shortened, and that is manifesting. Zygosity, it's a heterozygous or homozygous or compound heterozygous. Heterozygous is significant for your autosomal dominant or X-linked dominant condition, because only one mutation or variant would suffice, while in recessive conditions we'll be having 2 mutations to cause the disease. And the disease here is Rett syndrome, and it is manifested in X-linked dominant fashion. And the classification using ACMG criteria, that is American College of Medical Genetics, it is a pathogenic mutation. So this is how we go about the reading the report when we get. So as I said, when we can use, that was like example of a single gene, then we can have a panel. Panel means I didn't have any psychiatric case. I put this the lady who came to me as medullary carcinoma of thyroid, and then we found that we did the hereditary cancer panel, which looks for around 150 to 200 genes which are cancer causing in humans. So in that we found Rett. So this is again the example of a panel. Then now third, let's look at the clinical exome. This child came to us. So we have spoken about single gene, then panel, and then we go up and then slightly more genes. So that is called clinical exome. 3-year-old female, she had the classical features, which is of Noonan syndrome. So when I'm having Noonan syndrome, I know Noonan syndrome is caused by 50% of the cases of Noonan syndrome are because of PTPN11 gene, but then there are more than one gene in that. So even sequencing a single gene would not suffice here. So I want to do something more, but then at the same time, I know my diagnosis is more or less clear to me. So the clinical exome would suffice here. So when we did clinical exome, we found that the variant is in the PTPN11 gene. So this is the role of clinical exome. Clinical exome is means already known disease and genes. So around, as I said, 8,000, 9,000 diseases are known and their genes are known now. So that would be detected by clinical exome. So another, now coming to whole exome. Whole exome means, as I said, exome is a coding portion. So we are looking at the code problem in the coding portions of the gene. Whole means all 22, 25,000 genes which our body has. And then we are looking at that. So whenever I'm not clear, my diagnosis is not very clear to me. It's a non-specific. And maybe it could be this also, or it could be that also, some unexplained features which are not conforming to a particular disorder, then I would go for whole exome in that case. So here we did, and then found there are two variants. I want to show you this old report. It was uncertain significance, but then the upper one, ACTG1, has changed its significance over time, and now it is a likely pathogenic. And then we matched the features of the child, and there was some partial phenotype match. We went at the testing of the parents, and the child was carrying this, and so parents should not be, because parents were normal. So it was a de novo variant and parent did not have the mutation, and over time, and there was a match with the phenotype also, partial match, and then after that it changes significance to likely pathogenic, then we can take it as a causative. So what point I want to tell you about here, that whole exome should be considered when your diagnosis, it's you are dealing with odd, or maybe most of the labs have now moved from clinical exome to whole exome. So maybe clinical exome may not be available also. Yes. Do I have to take question now? Maybe later?
Yeah, later in the end, ma'am.
Later in the end, no? Okay, fine. So this is the significance of this, and I have not put any report, because as such whole genome, as I said, is something, the exome, what is exome not covering? It's not covering the intronic portions, the deep intronic regions. And I said in about 10 to 15% human genetic disorders, there will be mutation in that intronic portion can lead to disease. So when your suspicion is very strong and these are negative, then one can go for whole genome sequencing. So when you are dealing with just maybe a broad category of the neurodegenerative disorder, which most of you are seeing, you have done your clinical evaluation, you have gone brain MRIs, then you look for your dysmorphic features, hepatomegaly. You can classify based on these into various groups, lysosomal, Wilson's, if there are dysmorphic features, if we are dealing with paroxysmal. So this is the flow chart. But then what I mean to say, after doing the clinical, ocular, biochemical, your neural imaging, then the ultimate diagnosis molecule is established by the exome sequencing here. So the test of choice for these most of disorder would be, because these are single gene disorder and there are overlapping features. Overlapping features with most of the neurodegenerative may have, we cannot, though we see that yes, classically it looks like this, but then it can be this also. Then you can consider exome sequencing in your clinical practice when you're dealing with a neurodegenerative disorder. Okay, so word about the mitochondrial, because as I said, most of our DNA genome is in the nucleus of a cell, and small amount is present in the mitochondria, and this small amount is actually 16 kb, and it codes for only 13 proteins, 22 transfer RNAs, and 2 ribosomal RNAs. The 13 proteins which are encoded by mitochondrial DNA are involved in the OXPHOS, that is your oxidative phosphorylation, and basically respiratory chain. So most of you will find that the mitochondrial disorders affect the organs which are energy dense, because this mitochondria are the powerhouse, like your liver, kidney, muscle, and brain are the commonly affected, because there the energy requirement is maximum. What is the characteristic feature about the mitochondrial is that this is a maternal inheritance. It is passed from the mother. Heteroplasmy means there is a mix of the wild, that is normal, and the mutant mitochondria in there. One thing which I would like to emphasize, that though whenever see there's a mitochondrial disorder, mitochondrial, 2/3 of the pediatric mitochondrial disorders and 1/3 of the adult mitochondrial disorders are because of the mutations in the gene in the nuclear Not because the number of mitochondrial genes are limited, only 13, total 13 proteins they are forming. So if you are suspecting a mitochondrial disorder and you're only doing the mitochondrial genome sequencing, you may miss because they may be coded by nuclear gene. For example, here we have a child who is a 9-year-old boy. He's born out of a 3rd-degree consanguineous marriage. So consanguineous marriage, consanguinity means, but we know it's a blood relation, marriage is in a blood relation, and then it raises the chances of having an underlying autosomal recessive condition. So here, and the autosomal recessive condition would be mainly we are not looking at that in mitochondrial, we are looking at the nuclear genes. So there is an ID, there is symmetrical basal ganglia hyperintensities, which are actually leak syndrome like features clinically. And then we went ahead with the mitochondrial genome sequencing that was negative. Then we found that there is a, this one has MT-ATP6. This was a mitochondrial complex. This is an example of the mitochondrial disorder caused by mitochondrial genome. So MT-ATP6 is a mitochondrial gene and which is mutated and which is pathogenic. And so this is the child with mitochondrial disorder and with a mitochondrial DNA mutation. Another child here is a 2-month-old who presented with raised lactate levels, and initially it was thought of primary lactic acidosis or glutaric aciduria, and there was no positive family history. And here the mitochondrial genome was done, which was negative, and then we went ahead and did the nuclear genome sequencing. So in the nuclear, that is whole exome sequencing. So whole exome revealed here SDHA, and which is again responsible for leak syndrome, but this is the gene which is present in the nucleus, not in the mitochondria. So maybe if we have a phenotype of mitochondria, it can be caused both by mitochondria as well as nuclear. So you have to think of sequencing both if mitochondrial has come negative. So another, now after this, we have talked about the NGS whole exome. We have talked about the single gene, we have got the panel, we have got clinical, we have got whole exome. And maybe some conditions are still there where we need specific testing. So this is a boy who has this, you can see that this ADHD, he's on spectrum and with ID, and then he also had dysmorphism like prominent ears, there's a triangular faces. Basically, those who of you have seen maybe recognize it's a Fragile X syndrome. So whenever you're having a Fragile X syndrome, this is a disorder which is caused by triplet repeat expansion. The expansion here is CGG repeats. In FMR1 gene, there is a CGG repeat. So as of now, whole exome, we don't have a single test which can detect all the genetic mutations, all kinds. So we have to actually tailor. So if you are a Fragile X, you look for a particular type of PCR, that is triple prime PCR, to detect this triplet repeat disorder. Similarly, for your myotonic dystrophy or Huntington chorea, which is again a triplet repeat disorder, you will go for a specific testing that for those triplet repeat disorder, which will not be picked up in karyotype, in exome, MLPA, or all these. So this is, or we call it fragment analysis. So we have to know that the genetics behind the common disorders which we are dealing with, and accordingly we have to order the test. Maybe in times to come, in 5 years, we'll have one-stop solution. But then as of now, we have to choose the test wisely depending on the underlying condition. Similarly, if there is a Prader-Willi syndrome, as I said, or Russell syndrome, these are genomic imprinting disorders and there are methylation problems. So we will go for methylation studies, which are like MS-MLPA is one of the methods of that. So depending on the, and as I said, we have to emphasize that depending on the test, depending on the underlying problem, we have to define the test, and we have to know the different types of tests which are available currently. So just a flow diagram, if you're suspecting based on the family history and we are suspecting a neurogenetic disorder, then you have to select the test. If it is a autism spectrum disorder or multiple anomalies or unexplained neurodevelopmental disorder, go for microarray. And if you're thinking maybe I'm dealing, as I said, maybe Rett disorder, I will go for single gene analysis, or I'm suspecting Fragile X, I will for repeat expansion. And if all these come negative and you're still suspecting maybe it's a family history is positive and you're dealing, then go for NGS, go for your whole exome sequencing, and that may give you answer in further few cases. So the overall, if I talk about the autism spectrum, the combined yield of microarray and exome sequencing is around 30%. It's because, as I said, it's a polyfactorial condition. In about 60% cases, we don't have any genetic diagnosis yet. So a word about the adult onset neurodegenerative disorder. We know those common Alzheimer's, your vascular dementias, your Huntington disease or Parkinson disease. The basic problem in all these is there is a protein aggregation. Why this happening? Why this is happening? Because there is a disease synapse which leads to impaired proteostasis, vesicle transportation and autophagy, and that in turn causes protein aggregations, neuroinflammation and synaptic dysfunction and neurodegeneration. So and as I said, these are the conditions which are multifactorial polygenic. Polygenic means there are susceptibility genes, but mutation in one gene is not causing the disease, means it may or may not. So for these conditions, what we do, what are the studies? You can do genome-wide association studies that called GWAS. So GWAS is a method where you are comparing your cases and controls, and we are looking at the single nucleotide polymorphisms. And I said polymorphism is something which is present in more than 1% of the population. The variation which is present in more than 1% of the population, that is called polymorphism. So in GWAS, you'll find there are common SNPs. Suppose you're looking at Alzheimer's. So there are some SNPs which are seen in those patients who are having Alzheimer's vis-a-vis controls. So odds ratio will be relevant here. Similarly for Parkinson's also, or multiple sclerosis or amyotrophic lateral sclerosis, they're the conditions wherein you will have these are called susceptibility alleles or SNPs, which we are looking at. SNPs can help us that yes, we are finding this may be positive, but then as of now, treatment wise, the treatment remains limited as far as GWAS goes. So just to summarize how we have progressed from 2000 to 2022 and now in 25. So initially the problem was patient and family and the physician. We had limited tests and the testing helped us in diagnosis and the cascade family testing. As we moved on, we had better testing. We had CMA, we had PCR, we had targeted NGS, and that led to the prognosis and management. And now currently we have, with the help of genetic testing, we can have prognosis, treatment implications, cascade screening, and now this is between the patient, family, physician, geneticist, in collaboration with all of them. And just for any clinician, it's just about good phenotyping, just to have a knowledge about various tests, and adequate pre and post test counseling is required to understand it better. Thank you so much. With this, I have ended the talk and I can take any questions or any queries which are there.
Thank you so much, ma'am. It was really wonderful. Opened a lot of avenues for us to learn and understand. So over to chairpersons for the opening remarks. Then we take the questions in the chat box.
Thank you so much, ma'am. It was wonderful. Although we study genetics, since our high school days or even before that, it always remains an enigma. There are a lot of scenarios in our clinical practice. We always think that, will the genetic testing be useful or not? So with that context, I guess we will take up some questions in the chat. Dr. Robin, you want to say something?
Robin, you want to say something? I think his
All right, yeah.
connection is.
Go ahead with, there are a few questions in the chat box. I think first question was on, what's the function of mitochondrial DNA, which you actually dealt in the later part of your talk. Yeah, there are a few questions regarding, genetic testing with respect to ASD, autism spectrum disorder. So first one is from Dr. Gauri. She asks, are there any genetic tests to detect the severity or prognosis of autism spectrum disorders in a single patient?
In a single, okay. So as I said, the protocol which we are following for autism as of now, autism spectrum disorders, as per current guidelines, we are doing microarray. And if suppose I see a child who is actually conforming to a Fragile X, I would go for the Fragile X testing first, because that would confirm. In case I'm talking about autism, we can divide into syndromic and non-syndromic. When syndromic means like if autism can be associated with the NF1 also, it can be part of a tuberous sclerosis complex, it can be part of Soto syndrome. So when we're dealing with syndromic, the test would be different. When we are dealing with the non-syndromic, then the microarray you are doing, and I said the yield of microarray would be around 15%. generalize. Around 15% cases, you may get some microdeletion and microduplication. Once you find a particular microdeletion, then we know what is the implication of that microdeletion, how the children affected with microdeletion are going to do. Mostly we'll have a literature evidence that they are affected with the severe. Severity is usually defined by the clinical, as of all of you know that we are doing the psychological assessment, we are doing all the screening, and then further testing, ESA scorings, etc. And based on that, we are labeling the severity of a ASD. But yes, as far how severity can help, but severity can be defined by genetic testing, maybe because what kind of deletion it is there, or maybe once the microarray is negative, but you're still suspecting. Nowadays, what in my clinical practice, if microarray is negative, and especially couple is planning another pregnancy, in that scenario, more so in that scenario, because we cannot be sure that we are not dealing with a single gene disorder. So I generally go for whole exome sequencing and combined, because there are more than 150 to 200 genes which are, and more as we are sequencing more patients, we are finding a single genes which are implicated in autism spectrum. So the combined yield would be 30%, and depending on the underlying genetic, we can have some association of severity and the phenotype.
Yeah, if I want to frame it in a different way, I guess we have these parents, who come with, children, and now we diagnose them as ASD, and then they will be, very keen to know what happens to that boy or girl, later on. I think that was the context of this question. I believe there are more questions with respect to autism. Um, you know, Dr. Madhusudan says he knows of two instances where both the siblings have autism. Could this have been prevented by genetic testing before planning for the second child?
So there are two things to it, because there is a heritability factor to autism. Heritability factor, I said, so being a polyfactorial. So there is a concordance in the twins and other siblings also. If one child is affected, then there's chances having four around 8 increases. And if the two children are affected, the third may have a 30% chance. If it is a not purely monogenic condition or not dealing with the genetic part about it. So if there are two, as I said, one, then are more chances that second child could be affected. Yes, definitely the genetic testing should have been offered. And if it has been offered and we have found some variation, which is like maybe some autosomal recessive condition, if two are affected, or maybe two boys are affected, some X-linked problem, then if you have found, then we would have provided prenatal testing. Because through ultrasounds, we know we can only detect structure. The brain function cannot be detected. So only we can do prenatal testing by amniocentesis or chorionic villus sampling if we have found the mutation in the family.
Yeah. So what are the chances of someone having, when we get this genetic testing done, what are the chances of having these autosomal recessive conditions, and how can we do a cost benefit analysis of it?
Autosomal recessive means the genes, as I said, autism, we look at the CNVs by chromosome microarray. Those who are not having CNVs go for exome. And then obviously, as I said, for the syndromic, I'm not dealing with syndromic. You're having Fragile X and all, you're doing the testing, and that is there. And even if a non-syndromic also, microarray for a male child, we are doing Fragile X and we are going for exome sequencing. This is what we are following. And exome, we can have a mostly it's the autosomal dominant genes in our clinical practice, which we see. Recessive means parents would be carrier and child can be affected. Then the chances of recurrence would be 25%. While in autosomal dominant, child will be carrying one mutation, and most of the time this is a de novo mutation. De novo means it has happened during embryogenesis and parents are not having the mutation, or maybe parents are having the mutation. So it is important to do the parental testing also, to do the segregation, to know whether it is a de novo or inherited. And suppose what can happen if we find that if there is a child who is heterozygous and we have tested parents may not be having, that is, then it means it's a de novo and child is affected, the likelihood increases. And suppose one of the parent is having the same mutation and he's absolutely normal, he's not manifesting, then further you have to dig out that, see what is incomplete penetrance reported with this gene, means somebody carrying the mutation but not manifesting. Okay, so it may, a possibility, or it's just simply we are dealing with that the father has the mutation and it is simply passed on to the child, and it should not be a causative for the disease, any disease, that also. So these two things we have to see, if the one of the parent is having a heterozygous mutation with the child is having.
Yeah.
Hope it made sense. Hope I'm making it is clear what I'm saying.
I think in line with the same discussion, Dr. Abhishek, I think this is your junior Abhishek Pandey.
Abhishek Pandey, yeah.
In today's
Abhishek had, I saw later Abhishek has messaged me to slow down. Is it I was too fast? I mean, hope I made sense.
No, it was very well paced, I think.
Okay, right.
Abhishek must have found it too fast.
Okay.
So his question is, in today's world of legal implications, are we bound to test all children with autism as part of prenatal genetic testing where parents wish to have next pregnancy? I mean, this is, in
So
similar
this is
line.
something which is ethical, legal, and social implication. LC we are
He
dealing
also
with
asked,
here.
what is the yield in such cases?
Yeah. So yield, I told you the combined yield as of now is 30 to 35%. Combined yield of microarray facile X plus exome we're looking at. So combined yield we are looking at, and around 60-65% we don't have answer yet. Maybe the susceptibility genes, but then as I said, if we do GWAS in these children, autism versus normal, and then we find some single nucleotide polymorphisms, but then it would not be actionable. We can just for our study purpose, research purpose, fine, but then we can't take any action on that as of now. We are not going to do any prenatal testing based on SNPs because SNPs may or may not be causative.
Ma'am, first of all, a wonderful presentation. Actually, you taught us, it was a teaching class, how to read the report. That is very, very important, specifically that deletion part of the protein, because we used to just screen through, and the detailing actually has helped a lot. Ma'am, I have one case scenario. One of the kids came, ma'am, with schizophrenia and epilepsy, and we found that she had basal ganglia calcifications. Was with past disease in AIMS Delhi. Subsequently, we found that brother also had a similar scenario, but brother is functional. He also had the same basal ganglia calcification. We got a referral from neurologist in a center here, and then he suggested a genetic testing subsequently to see things further. So, ma'am, what specific test we'll send in this scenario?
Yes, as I said, because here you're dealing with a epilepsy, you have a
Yes.
seizure, and
Yes.
basically maybe I guess that's a regression you are dealing with. Along
Yes.
with the seizures, there is a condition which is neurodegenerative with the basal ganglia and dystonia or anything which is there, some dystonia, no extrapyramidal movements or something.
Which are controlled, psychosis which is not controlled, and family history of basal ganglia calcifications. And brother is functional, she is not functional at all.
Oh, okay. So have we looked at the coppers also, copper levels?
Ma'am, yes,
Copper.
yes, they have actually idiopathic basal ganglia calcification there.
Idiopathic basal ganglia calcification. In that case, I would because I say most of these are monogenic, and whole exome sequencing would be something which will be in place here.
Okay.
If I may, I think one of the issues is we are talking about genetic testing and we are talking about psychiatric disorders. The problem with psychiatric disorders is many of the disorders are actually collections of symptoms. So they are not true diseases to the core, if you can say. They are collection of similar sounding symptoms. So if you talk of one diagnosis, there will be many flavors in it. So you take one disorder, you will find sometimes genetic testing would be positive, sometimes it would be not, because that entity is not biologically homogeneous. A lot of heterogeneity leading to similar sort of presentations on the surface, but in the base, it is different. So I think my co-panelist, the psychiatrist here, your comment on this.
No, we are seeing one thing I would like to add here, because there are so many neurometabolic conditions in adults also, but not only childhood, adult onset, they can present with neuropsychiatric manifestations. So whenever that's what I'm saying, unexplained neurocognitive impairment, neuropsychiatric manifestations, one has to now think of the underlying neurometabolic or neurodegenerative process that is there.
To actually just circumvent what Dr. Aleem sir has said, in addition to this, because Sai is also here with us. Ma'am, you said that one of the indications for doing a genetic testing is dysmorphism. So, Sai, but we have read and practiced minor physical anomalies. So we have given it a name as minor physical anomalies. So MPAs, but we wouldn't generally go for genetic testing. So now, after all this, we are finding in adolescents with psychosis with MPAs in the umbrella of dysmorphisms, we are going for genetic test. So your take on it, Sai?
I guess it's MPS are there in all of us. What is important for us to know is what are the higher number? When do we call it as higher number? So, any psychotic disorders or, which can be explained neurodevelopmentally, we have something called 5 as a number, 5, 6 as a number, and then more than that. I guess, then we'll have to look at panel of genes which are neurodevelopmentally implicated. I hope if I'm not getting it wrong that way, ma'am can tell me more on that. So if what are the genes that are neurodevelopmentally implicated in later onset of psychosis? I think then in them we can go, but still I'm not sure. I haven't.
Sai has given a quantification to it. The
Yeah.
dysmorphisms, if they are a particular number, then we should go about it. Or ma'am, we should go for the genetic testing if there are subtle dysmorphisms like cyanophres and all present also.
Dysmorphisms, yes. I mean, there's an obvious dysmorphism because subtle may not be picked up by everyone. But then there is, as I said, those four things when you suspect broadly. That is presence of dysmorphism. There is an unexplained neurocognitive impairment. There is a family history which is positive, or presence of a multiple congenital anomalies. So Occam rhesus hypothesis. I mean, unifying explanation for multiple problems. So if we should not be work in silos. If you are a patient, and I always tell my residents also, if a patient is making so many visits to multiple specialists, then you have to think maybe there is something which is common, which is causing multiple manifestations, and look for that particular that unifying etiology in that. So these are some of the conditions or situations where we have to think. But then having said that, as more and more we are testing more and more people, more and with the neuropsychiatric manifestations, we like, suppose about the Tay-Sachs disease, which is common in Ashkenazi Jews, but then one of the manifestations of Tay-Sachs is a late onset, the only psychiatric issues or gait ataxias, Niemann-Pick for that matter. So many lysosomal storage disorder, neurometabolic conditions have got late onset neuropsychiatric manifestations. And we are seeing obviously early onset Alzheimer's, Parkinson's also have genetic component. Recently we sequenced a whole family having early onset Parkinson, and they had genetic variation which is there and segregating with the patients. So early onset disease which is presenting earlier, more severe, multiple affected family members in, I'm talking about the psychiatric conditions also, then these may be the pointers towards genetic testing.
So I think we'll discuss more about clinical implications in later sessions.
Yes. Today was that's what I was thinking, because there'll be too much to discuss in one go.
Yes.
Yeah, and then one knows what is exome sequencing, what is MLPA, what is microarray. So what the basic idea about the testing, and now it can be tailored to psychiatric, specific.
But if can
Vishal, please carry on.
just one, I want just for the basic understanding, very basic for all of for now who doesn't understand much genetics. When will be the day you see, because you've talked about a lot of tests, where testing will be as easy as testing CBC, LFT, KFT, we ordering for patients?
In fact, we have made good progress in that direction. That is the thing. I mean, if 2016, when I was in SGPGI doing my DM, that time the cost of a whole exome sequencing was 35,000. And today it is 12,000, between 10 to 12. So 10 to 12 is still affordable. Most of the government institutes have started doing it. In fact, in armed forces, we have started doing it. We have platforms and we are doing in-house. So we are able to do. Only thing, so that is mostly reasonable now, and then in times to come it will be much cheaper because with the availability in most of the government setups that is there, and it's okay. I mean, obviously it cannot be as cheap as HB, TLC, DLC, but then it is something which we can afford. MLPA, the cost of MLPA is 5,000.
Okay.
Yeah, so the cost wise, microarray is around 10 to 12,000, and a microarray was used to be 25,000 earlier. Now it is half the cost because of the multiple people who are offering it now, and then with the government this thing coming up, it is affordable now.
So Dr. Madhusudan, parents want to know which one of them is to blame for a child's condition. Is there an answer?
And that is the problem, which usually, but I mean, all the more reason we have to tell, because most of this kind of society we are living in, and then always they feel one of two people that they live in guilt conscious, and especially if it is an X-linked condition, if the mother, but since the X-linked conditions come from the mother, I'm always as a geneticist skeptical about telling. So basically, when we are diagnosing more, if it is a de novo, we can say you are not to be blamed. I mean, this has happened on its own during embryogenesis. Parents also, feel relieved that, okay, we are not responsible. They feel the guilt conscious is removed. So the testing and then establishing a de novo variant First of all, identifying the variant, then identifying that is a de novo, it helps. And then obviously it's a de novo, the risk of recurrence would be very low. That would help in the recurrence and then further counseling, reproductive counseling also.
But to establish de novo, we need to test the parents. That is
Parent,
what-
obviously, de novo means it is not present in the parent. So whenever
Yeah,
you're
to
finding,
test the parent to
especially
establish.
yes, for an heterozygous, and when it is a homozygous or compound hit means two variants are there. That is, we are dealing with the autosomal recessive condition. In that case, parents would be carrier and carriers are unaffected. Each parent would be carrier of each variant. क्योंकि दोनों क्रोमोसोम पे चेंज रहेगा, एक एक म्यूटेशन जो एक एक पेरेंट से आया है. The child would be affected in this case for a recessive disorder. For a recessive disorder, carriers are usually asymptomatic.
Okay.
Yeah, I think Dr. Robin, if
Yeah, thank you. Actually,
some
I got
questions in the-
disconnected
I'm sorry.
between. But thank you, ma'am, for this wonderful talk. And I take this talk in a different way. I consider that being a psychiatrist, we consider ourselves very much apart from all these things, and we are more into syndromal based or symptom based diagnosis. But I think learning more about genetics will give us better picture of psychiatric illnesses. So our patients always ask this thing that why this is happening, and we generally say it is unknown etiology. But I think in future we will be in a better way, but we will be in a better position to answer them that why this is happening. And I think being a psychiatrist, we should take more interest in this topic. And it will be good for the subject also.
Yep.
There are some more questions in the chat box. I'll
Sai, please cover.
Yeah, Dr. Alka, are there, I mean, she has a couple of questions on mitochondrial DNAs. Are there chances of mitochondrial disorder proportional to maternal aging? I guess, I think in terms of advanced maternal age is what she wants to ask.
So basically, all the mutations can have, because chances of having mutations may increase with the aging. That is there. But then, as I said, what happens in the maternal mitochondrial, that total something called as I said, heteroplasmy and homoplasmy, something called bottleneck hypothesis, that you are only homoplasmy is either you are having a mix, heteroplasmy is a mix of the wild and then mutant. There's a mutant mitochondria and the normal mitochondria. And then homoplasmy means either you have a completely wild, that is normal, or you have a fully mutant one. So in a homoplasmic conditions, usually people develop manifestations, and heteroplasmy depends on how much is the heteroplasmy level, how much is the mutant, and in that particular tissue, mitochondria in that particular tissue, how much it is there and how much in that particular tissue it is mutant. So it's a little complex genetics of mitochondria. But as far as the aging part is considered, it would be generalized because of the exposure to the those environmental toxins, radiation, and a lot that every day that causes wear and tear mechanisms. So that would be generalized for all the DNA
Recently studying something called, Grimmage, and there are these, DNA methylation clock. Methylation clocks.
So that is, we are talking about epigenetics.
Yeah. So I think with respect to epigenetics, there's one question here, or that even in cases of monozygotic twins, if both have psychosis, there are different presentations. I think epigenetics is the answer here.
Yeah. So that is what nature versus nurture. And then because if it nature wise also monozygotic would be sharing the DNA 100%, and I would say nurture means they're in the same environment, but there are something genetic modifiers that may differ. Okay, and then they can, that you said, epigenetics, then the methylation, abnormality, methylation changes which can be there, and how the histone is packed in these monozygotic twins. So that is a lot of studies are going on in the field of epigenetics. Also, in fact, we have a whole conference on epigenetics we are organizing on 1st and 2nd November in Delhi. I would share the brochure with Dr. Shobit. If you can pass, somebody is interested, then they can join.
You spoke about the MLPAs, with respect to DNA methylation. Is it?
Yes. One of the methods of doing methylation is specific testing. So what is epigenetics means? One of epigenetic mechanism is the silencing of the gene. One of the methods of gene silencing is methylation. Methylation is basically addition of a lot of cytosine and guanine residue. In that CG, we call it CG residue. So that can be studied by MSMLPA, one of the methods. So like those Prader-Willi and all have heard the these are in genomic imprinting disorder. Herein they have one copy and that other copy is silenced. one copy is already silenced, another copy is now mutated. So to study that, we are doing the methylation specific testing.
So, ma'am, let me say a hypothetical situation. Suppose in psychiatry, we are not dealing usually with single gene disorders. We are having multifactorial
Yeah.
poly. So, suppose you have to deal with a multifactorial with many genes involved. So, how do you start as a geneticist? What is the level of testing? First test kaun sa denge? What next? What next? So that you can reach out. You have already told
So
it in
that
the
depends
presentation.
on what
Just a summary here.
Yeah. So that depends on the underlying, suppose you're dealing with obsessive compulsive disorder or mood disorders only. So here I would place, because vis-a-vis, if I'm dealing with the Parkinson's features, which is causing mood problems, or Alzheimer's, which is causing psychiatric issues, neuropsychiatric manifestations. So the purely OCDs, MDs and all will have a lesser component of monogenic this thing, but they can have, as I said, the SNPs. If there is a mood disorder which is running in the family, so maybe there is a polymorphism which is there in that family. So for that, the genome we already have, like for schizophrenia and all, we have genome-wide association studies, and we know there are some SNPs, that is your single nucleotide polymorphism, which can be seen. So that can be studied, but I said the action-wise, we cannot take much. We can just tell them, yes, there's something, there is susceptibility gene which is running in your family. That is what. So for depending on the underlying psychiatric problem, knowing the heritability index in that, then like if I'm dealing with early onset Alzheimer's, or for that matter Alzheimer's, we are knowing have some are monogenic in nature, I will go for whole exome. Or if we are only dealing with the spectrum of mood disorders or OCDs, etc., bipolar disorder, then I would be going for more of a GWAS if I can offer. But GWAS is something which is very tedious thing. It has to be taken with the institute and all, but then may not be implicated in a single clinical practice.
So whole genome and GWAS are two major things.
Whole exome and GWAS. Yeah. For use
Correct.
for psychiatric, maybe in next, if I'm talking or somebody else is talking, they can cover about psychiatric, because as I said, this would be a talk about the basic testing and understanding the basics of the things, and later it can be tailored to psychiatry.
Ma'am, how to ensure that like accuracy of test, because we are working in private sector. That's to Dr. Sai, who can furnish plethora of tests on his institute's behalf. So if we are on the side of sending a test, maybe Sanger could have picked it up, but your whole exome doesn't pick it up. Or so, ma'am, how to ensure that the liaising with geneticist is very, very important, I suppose, ma'am, in this case.
Yes. You have to liaise, you have to consult. Don't do everything on your own, and not in silos, because yes, we have to see the quality. We do run, whenever we get the data, there is a quality check. We have to see maybe the particular genes of interest, which one of my genes of interest are covered or not. Coverage means, coverage and depth are two things which we are looking at. Coverage means how from one end to other end, it has been covered or not, fully covered, the gene. And then secondly, depth means how many times it has been gone, repeated covering, so that we like particular sequence, agar maine bola 20x means 20 times it has been read, so that the chances of more you read it, more the chances of not missing the mutation. So this is called coverage and depth. So you have to see that, first of all, you have to know your problem, the clinical problem, then the test with what genes you are dealing with, and those genes were covered or not properly. Maybe that would be, to some extent, can give you some answer.
Okay, ma'am.
There are a few more questions, ma'am, a few of them. Dr. Madhukar Katyar, he asks, so if there are two parents with dementia, now we're moving on to, from childhood to age, dementia, one is with Lewy body, the other one with, say, Alzheimer's. So as an adult, what are my chances of having, any of these dementias?
Okay, so again, a multifactorial complex, both the parents are affected. About roughly, we give empirical risk of recurrence, and we have the empirical risk of recurrence for these conditions, which are available, maybe around 30% which is there, I remember vaguely. But if you want, then we can talk about, I mean, specific is more or less, if somebody is having one Lewy body and one is having Alzheimer's, and both are multifactorial, as we are taking as a polygenic. So the chances would be around roughly 30%. This is empirical risk of recurrence, which we are giving.
I think we should end it now. We have already reached.
Yes, we have covered all the questions, and Dr. Aradhana is constantly making an effort for us. So I think we have covered all the questions. Yes.
And I think most of the testing question will come second time when we're talking about disorders.
Most of the more practical things in the
There's
next
one-
session.
There's one question which has been repeated, this is by Dr. Akshay. Maybe if you permit, we'll take this question and then.
Yes, yes,
Please.
please. Hmm.
Yeah, this is how to diagnose the case of 17-hydroxylase enzyme deficiency type of congenital adrenal hyperplasia is the question.
So ultimate diagnosis by the molecular testing only. I mean, this is 17 hydroxylase. The most common is 21 hydroxylase deficiency because I know, but the 17 and 11 beta and 3 beta dehydrogenase and all those will have little slightly different manifestations. And we have got different genes for this. So you can go for the clinical exome sequencing or whole exome sequencing in this case to have one of these methods can be done if you're very sure about 17 alpha. But I said there are always overlapping features. So single gene would not help. Better to go for a more wider test.
Yeah.
I think I'll just a brief one, few second scenario where such excellent resource person like Aradhana ma'am can help us. But a child came with intellectual disability and OCD. Actually, brother suffered from intellectual disability and psychosis. Both had alopecia areata. And subsequently, we did a genetic testing of the patient who contacted us. We found an ATP mutation. Subsequently, we got in touch with madam and we were asked to do it for the brother and also for the parents to establish whether it is de novo or it is. So I think this is something which can solve this question of why in families it runs in families. So I think a very important context is liaising with the
Yes, obviously, because it's a multidisciplinary approach, and always for any, I mean, whether it's a psychiatry, whether it's neurology, all of us have to work together to get at the common, page with this.
Thank you so much, ma'am. I think we should start winding up now. So comments from
Yes.
chairperson, then to Tofan sir.
Yeah, I mean, it was a wonderful talk, and as you said, this is start of the series, so it's a great beginning, and we look forward to your next classes. Thank you, ma'am. Dr. Robin.
Thank you, ma'am, for enlightening us. And now I think I should read properly. So thank you for sensitizing on such an important topic. And hope in the coming series we can get a lot of new things into it. Thank you.
Thank you so much. Thank you.
Tofan sir, over to you.
Thank you, Dr. Aradhana. Your talk was very nice. Actually, our psychiatry started in late 70s and early 80s when genetics was not as much developed as of now. But I had the pleasure of meeting Chuang and Unekur personally in those days. And what I find your talk is very much educating. It was a class. But I have got 2 or 3 queries. What is the scope of violation of ethics and ensuing legal problems? Do you know anything over there? For example, confidentiality, privacy, and autonomy of the patient may be violated. And is there any legal issue in our country?
So there are, as far as I said, that we are doing all the testing with the proper pre-test counseling. That's called pre-test and post-test counseling. These are the 2 things. Pre-test means before the test, we are telling this is the type of test, this is the cost of the test, and this is the yield of the test. Means what? Because at times patient feel that I'm not talking about armed forces. Where armed forces they are not paying, so that is different thing. But where outside they are paying, they feel if I'm getting a test of 20,000, it means that would solve all the problem. So to tell them the yield of the test means after doing this also there are chances that कि autism में अगर हमने microarray किया तो 10 से 15% में मिलेगा, still it would be unanswered and then we can go for some other testing. So this has to be told beforehand. So this is there. And then secondly, sir, once we have spoken about all these things, then something called, when we are doing the exome sequencing, we have called something called secondary findings. We have now guidelines. So we always get a signed thing that if there are secondary findings, these secondary findings are related to the mutations in the genes, which is causing cancer, which is causing cardiomyopathy, which are actionable. मतलब हम कुछ और देखने चले थे और साथ में कुछ और मिल गया. So these things have to be told, because whenever we are looking at the complete genome or exome, we may find something which we're not expecting. So some secondary findings we have guidelines and we report those secondary guidelines also. And thirdly, as far as X-link, another social ethical issue comes with the, I mean, so ethical means more of a social issue, I will say, about the X-linked conditions wherein mother is a carrier and a mother has passed, or mother asymptomatic mother has passed to the sons, like a typical case of Duchenne muscular dystrophy. So we look at as a geneticist, I look at the sensitivity and the background of the family, and accordingly I'll tell them whether mother is a carrier or not. We don't reveal it. If they feel that the mother would be boycotted or stigmatized in the family, which happens, which has been seen, which if they are educated, they understand the problem, then we tell them, but then not as obviously as we tell about the autosomal recessive condition, wherein both the partners, if the both partners are carrier, they are okay with that. If the mother is a carrier in X-linked condition, there is a social issue with that. So we are little, conscious while and cautious while counseling the families with an X-linked condition. And as I said, secondary findings also we are covering, and we are telling them beforehand that something may come up. So it's a good pre-test and post-test counseling about the test, about the limitations and the utility of the test is required.
Thank
That
you.
means there is no legal dispute. Okay, we'll discuss later on. Because, but to the best of my knowledge, the Myriad Diagnostics had patented 4,000 and odd genomes, which was disqualified by the US Supreme Court about 10, 12, 15 years ago. That is misutilization of the data that ensues.
Yes, so the privacy of data and handling of data is again a big thing, which we are not discussing. I mean, in the clinical
that is
parlance.
also important. Okay, thank you very much, because you have made it very simple.
Thank you, sir. Thank you.
Thank you. Amrit, wind up.
Thank you very much.
Yeah, when I looked at the topic, I thought I'll find somebody who's very serious, very grumpy, teaching us about genetics. Because whenever in college we had somebody teaching us genetics, it was somebody like Dr. Alim. So it was very refreshing.
You've been
It
pulling
was very
Alim like Dr. Alim like anything I've been seeing.
so,
So
but
I
this I
have met
never
Dr.
expected.
Out of all of you, only Dr. Alim, I have met him twice in his clinic, and his demeanor is very calm and composed. And my mother तो maybe मिलके शायद ठीक हो गई थी आधा.
वो होता है ना, अलीम को हम लोग कॉलेज में एक चीज बोलते थे, वो he's a CPU without a monitor. So brain is working overtime, face does not work at all. So anyway, thank you so much. It was beautiful. And thank you, Dr. Rabin and Dr. Sai. Dr. Sai गायब हो गया है, रसोई कर रहा होगा genetics पढ़ने के बाद. And thank you. It was very a difficult topic. I'll tell you, it's very difficult. It's really a tough topic.
But then
And
that's
to
what I
make
was trying to make
yeah.
more, simpler.
Yeah, simpler.
Try to
इतना
do
simple
that.
करने के बाद ही अलीम को बहुत difficult लग रहा है, और रात को फिर पढ़ेगा. So thank you so much. Thank you everybody. Good night.
Thank you.
Good night,
Dr. Alim, चलिए happy Eid. कल Eid है ना?
Yeah. Thank you.
Yeah, चलिए wish you a very Eid Mubarak.
Thank
in
you.
advance.
Okay.
Thank you, madam.
Bye.
Thank you.
Bye.
And happy Teachers Day, everyone. Happy Teachers Day.
Yeah.
Yeah.
Happy Teachers Day.
So you are a teacher today. Yeah. Thank you.
Many we are all learning. It's a two-way process.
Obviously.
Yeah.
Thank you. Ending the meeting now.
Thank you so much. Thank you.
Thank you, everybody.
Good night.
Good night.
Good night.